Okay. So now I can change from organizer to presenter. My tag. So yeah, my name is Luca Privileggio. I come from Rovinj, from Croatia, and I finished my studies in nature conservation here in this building at FAMNIT. But I did my master thesis at the Center for Marine Research in Rovinj. And it's entitled, it's a long title, Estimation of Marine Contamination based on the potential toxicity of water, sediment, biota in the Northern Adriatic Sea. So let's start. So introduction. As you may know, the marine ecosystem is constantly under a big influence of both natural and mostly anthropogenic pressures. When I'm talking about these pressures, I'm talking about contaminants that go into the sea. And contaminants are interesting compounds because they tend to change during time. They tend to change and they tend to maybe merge and become more toxic or less toxic. They're a bit complex. And it's not enough just to see what contaminants are present. So you make a list of what is present in this sentence site. But it's important to see this interaction between them. And it's important also to see how they distribute differently in the water column and how they are accumulated variously in the biota and sediment. When I'm talking about biota, biota are living organisms. Okay, so fish, marine mammals, like everything. And the toxicity assessment is a well-established tool to achieve this, to see the contaminant accumulation, the contaminant behavior, and the potential negative effects on the marine environment. So what were the aims of my study? So it was to determine the potential toxicity of 3 different matrices or environments. So the seawater, sediment, and biota. For biota, I used like the mussel, Mytilus galprovincialis, the one you eat with pasta. So at each selected environment, so sampling site, and test correlation among those 3 potential toxicities. The second one was to determine the ability of this potentially toxic environment to affect the mussel anoxic survival. You'll see about that later. I'll explain a bit better. And the third one was to determine the toxic potential is related to the sediment type. Don't worry, I'll explain as well this. So let's start. As you can see, I did something. I collected the samples myself. So it was mainly in the Northern Adriatic Sea. Pointer is here. So mainly in Rovinj, in Limfjord, Pula, and in Kvarner in Bakar. Here we got like the zoom in pictures. Mostly are urban areas. And yes, I had to dive there. So what don't you do for science? I mean, I wouldn't take a swim there if I didn't need to, but yeah, okay. So what did I do? So I used this Microtox Bio essay on this very modern computer, as you can see, with Windows 95. So I got these organic extracts from the mussel, seawater, and sediment. And I tested these organic extracts on the bacterium Alivibrio fischeri. I was still looking for this number, this value, AC50. This is the estimated concentration expressed in milliliters or microliters of the specific organic extract used to diminish the bacteria's luminescence by 50%. In a few words, I was trying to kill the bacteria to see how many microliters or milliliters of extract do I need to kill 50% of all bacteria. And this is the toxicity value that I was looking for. So not a list of compounds, but a value. Next, this is the mussel anoxic survival or SOS test. It's not SOS because they need help, but because it's stress on stress help. Basically, you put mussels in trays and leave them to die. You then check their survival time every day, and you're looking for this LT50 value, so little time expressed in days, at which again 50% of the muscle specimens were dead for every sampling site, of course. Then for the granulometry, I used the sieves, like regular sieves, to determine the grain composition of the sediment. So if a sampling site would have a much higher percentage of gravel or sand or silt and clay, to differentiate them. What did I get in my study? So for results, I won't bother you with technicalities about graphs and everything. So I used this Spearman's rank correlation and the principal component analysis, also known as PCA. So I was looking for, once again, to see if there's any correlation between the potential toxicity of seawater, sediment, and mussel in this case. So basically, if I go to sample, I wanted to check if I sample seawater and I get results that is potentially toxic, will I get the same results if I take the sediment and if I take the mussels? The answer is no. They differentiate between them. So I found no correlation between them. So it's important to take samples in all three matrices and not just one, to get a full picture of the environment. As well, I checked is there any correlation between the potential toxicities of these three matrices and the SOS test. So this was the mussel survival, to see if the potential toxicities in that environment would affect the anoxic survival. This survival, as you may know, mussels survive outside water, like for 7-10 days. But no correlation were found. So it depends really on the specimens, on the site, and everything. So to also, once again, explain why they differentiate these three matrices. So mussels are living organisms. That's the first one. It depends on the species. It depends on their diet. It depends a lot on the season, as we know when you eat them as well. It depends on their digestive physiology. But the most important thing is to remember that their metabolic rate, because as they are filter feeders, they so filter the seawater to feed practically, and they automatically introduce a lot of toxins and contaminants into their body, and then they need to metabolize these contaminants every day, constantly, and it depends how well this metabolic rate works for them. So it depends a lot on these factors. Then we have seawater. This is a specific spatial and temporal point. What that means? If I take a bucket of water now, and I check the toxicity of that bucket of water, in half an hour, I take the same bucket of water, but it doesn't need to be the same toxicity. It depends a lot on wind, tides, currents. It's an open system. And then we have the sediment, which is a sink area. Everything that we can find in the water column eventually will sink to the sediment. And it's really important to understand, so there's an upper layer and a deeper layer. The upper layer, you can find there the novel contaminants. And in the deeper layer, you can find the older contaminants. So it's your decision how far you'll go. If you go like 1 meter, 2 meters, 3 meters, you can go 100, 200 years ago. So when I was doing my research, I just grabbed like the surface, like 1 centimeter max, to get like this toxicity in recent, let's say, recent year at least. Then we have here again some results. Again, the principal component analysis and a linear correlation. So now I was trying to see if there's any correlation between the sediment toxicity, so just sediment toxicity, and the granulometric composition of the sediment. In a few words, if I found in a sampling site that there's a higher percentage of gravel, maybe I presume that automatically I will find a higher toxicity, or maybe with sand, or maybe with silt and clay. I found no correlation. So it doesn't depend on the granulometry of the sediment. It doesn't mean anything. It depends a lot on the sampling site. Maybe it's in front of an industrial zone. Maybe it's, I don't know, under some other anthropogenic pressures. So it will depend. And to conclude my presentation, what's the take-home message for everyone? Considering all data present in this research, it is crucial to establish the potential toxicity of all three matrices. So sediment, seawater, and biota, in this case, the mussel. If you want to have a realistic picture of the sampling site, and if you want to protect it, if you want to get this good and exact environmental status. Second one is that this microtox bioassay, so that computer with Windows 95, it's really good, although slow, but and it's very convenient as a, let's say, a primary test. So you just grab a sampling of, whatever you want, sediment. So I mean, you need to take all three: sediment, seawater, and biota, and then you get like an idea of how is the sampling site. And then if you want, you can go and check exactly what contaminants are present there as well. And then you can go and take more actions and measures to protect that area. And that's it from me. And thank you very much.